third person, instead of Amy, we're gonna go with the Marco Carpenter. He's from the Children's Hospital of Philadelphia, so CHOPS. And his title will be the unique feature of the fetal brain permit non-viral mRNA delivery. So still RNA word.
And a little bit of spatial transcriptomics as well. Hi, I'm Marco, for those of you who I haven't met. I'm at the Children's Hospital of Philadelphia at the Center for Fetal Research, and today I'm going to talk to you guys about some of the unique features of the fetus that permit LNP-mediated mRNA delivery to the brain. And so gene editing really represents, there's an unprecedented opportunity to cure monogenic diseases. There are over 7,000 rare diseases, 80% of these are monogenic, and 83% of these diseases affect the child. And of those 83%, 90% of those comprise the CNS, and thus there really is a critical need for early intervention specifically for CNS disorders. And just to go back to the basics, underlying these genetic diseases is our genetic code, in which a single nucleotide change can cause or lead to disease. And so I use gene editing tools such as CRISPR-Cas9 to perform gene editing, and traditionally, traditionally we can either cut out these pathogenic areas, which leads to normal function. Today I'll talk a little bit more about a base editing approach in which we can make single nucleotide changes such as A to Gs or Cs to Ts, which again leads to normal function of the gene. And then in the future, and really what's, where the future is going, is that we can completely rewrite genes, pathogenic genes, and restore normal function. And so the fetus is really a formidable target for gene therapy. Not only can we cure the disease before the onset of irreversible pathology, but also there are a number of other factors that permit efficient gene editing. For one, there's open fetal circulation, which allows for direct injections to the fetus. There's a fetus of small size, which allows us to give less drug, making them potentially cheaper. There's also in the context of gene editing, we have access to stem cells, which make our drugs more efficient because those gene-edited cells then propagate. The immune system is also naive, and so in certain contexts, we can give higher doses of drugs to avoid an immune response. And really the focus of my talk today is that some of the biological barriers, such as the blood-brain barrier, are more permeable. And so at the crux of gene editing are proteins, such as CRISPR-Cas9, that we can deliver via mRNA using lipid nanoparticles. And so a lipid nanoparticle, most of us in this room probably have one in our bodies or have taken one before, is made up of 4 components, which is an ionizable lipid, a helper lipid, a cholesterol, and a PEG lipid. And through microfluidic mixing, they self-assemble and can encapsulate mRNA. And once encapsulated, this mRNA is protected from degradation once administered into systemic circulation. And once these lipid nanoparticles reach the cell, it initiates transient protein expression in the cytosol. But LNPs have been predisposed as well as optimized for liver targeting. And so the potential pathway to the brain really involves systemic. I'm going to move over here because there we go. Once they are inserted into the systemic circulation, the serum proteins bind, which allows them to interact with receptors on the endothelium of the blood-brain barrier. Once they interact with these receptors, they can be endocytosed into the endothelial cells, under which they go 3 major fates. They can be transcytosed from the endothelial cell into the brain parenchyma, where they can target neurons and microglia, which are important for a lot of these CNS disorders. They can also undergo endosomal escape, in which the mRNA is released into the cytosol, leading to protein expression and gene editing of the endothelial cell itself. But where the majority of this drug goes is to the lysosome, where it is degraded. And we know that less than 1% of the mRNA is actually released into the cell, and we know that probably even less of it is actually getting into the brain. And so really there's a lot of improvement that we can, to make these drugs more efficient. And so to screen LNPs for brain delivery, we use a very simple model in which we can deliver mRNA Cre or Cas9 with guides to cut out the stop cassette, and this leads to GFP expression. And what I'm showing you here is just some IHC of an adult brain following a fetal injection, which shows nice GFP expression in the brain. And so my first project in the lab was to really understand how does the ionizable lipid component affect cell type specific drug delivery. And so I used the lead ionizable lipids in the field, and I then formulated them with standard formulations of cholesterol dope as well as PEG. I then encapsulated mRNA for these studies, and then I used these LNPs and I administered them to either the fetus, the neonate, or the adult. And then for long-term editing studies, I looked at the adult, and then I used flow cytometry to quantify cell type-specific gene editing. And so using this technique, flow cytometry, we looked at endothelial cells, microglia, as well as neurons. And we tested our 3 LNPs, which have a different LNP or a different ionizable lipid component. And what we found is that in endothelial cells, to our surprise actually, that a lot of the LNPs actually can reach endothelial cells. And we did see some differences in how LNP composition can affect cell type specific delivery. But really the interesting thing was that only following in utero injection or fetal injection do we see that microglia as well as neurons are targeted, which gave us a little bit of hope that potentially if we perform in utero injections that we can target some of these important cell types. And so preliminary, we conclude that not only does LNP composition mediate cell type specific drug delivery to the brain, but early drug delivery facilitates diffuse targeting in the CNS. And we next kind of just ask, can this be applied to treat disease before the onset of irreversible pathology? And so I first started with, I'm working on a number of diseases, but today I'm going to talk to you about spinal muscle atrophy, which is a very common childhood disorder, which is caused by a deletion in SMN1. And this leads to rapid neurodegeneration and early death within the first 2 years of life. And we kind of know from the literature that we can correct this disease through base editing of a homolog gene, SMN2. And again, we're using base editing here where we're converting an A to a G, which leads to normal protein function. And so when we look in the liver where LNPs are supposed to go, we saw what we expected to see, right? That we see some A to G conversion, about 30% following fetal injection that was much higher than what we saw in the neonate and above control levels. As expected, in the liver, we see that protein levels are increased following fetal injection. But unsurprisingly, and it's kind of disappointingly, when we looked at the when we looked at the brain, we saw very, very low levels of editing, around 3%, and there really was no difference between fetal or neonatal injections, although it was above that of controls. We saw no differences in protein expression in the brain. But what was exciting is that we did see some survival advantage for our gene therapy. And so what's shown here in controls in this SMA mouse model, they succumbed to the disease about 5 or 6 days, and we saw a nice survival advantage for those that were injected fetally, and then we saw an intermediate survival advantage for the neonatal injected mice. And really this indicated that there not only did our therapy work, but also potentially that fetal injections are more efficient than neonatal injections and may be able to cure the disease or mitigate the disease prior to the onset of irreversible pathology. And really what this also showed us is that we have a lot of work to go and there's a lot of room for improvement. And so how do we do that? And what makes lipid nanoparticles so cool is that we can actually not only change the formulation, so insert different ionizable lipids or different helper lipids, but we can also add conjugate antibodies or specific peptides to the lipid nanoparticles, which allows them to target specific cell types. And so my hypothesis is that if we can increase targeting to the endothelial cells of the brain, potentially we can increase transcytosis and targeting of the neurons and microglia in the brain. And so I turned back to kind of my expertise that I learned as a grad student, and I used single-cell sequencing to identify candidate genes and putative targets of LNP uptake. And so in this data set, I integrated a data set across the developing as well as the fetal brain. And so we have like a fetal, an in utero component, a neonatal component, as well as adult cells, and then isolated endothelial cells, and then looked at a bunch of putative endothelial target genes that I just identified from the literature. And I'm going to focus today on 2 receptors, receptors X and receptor Y. What we found was that receptor X shows a pattern really consistent with what we were seeing in terms of transcytosis across the blood-brain barrier, and that it showed some expression in endothelial cells early in life that waned later in adulthood. And then another receptor that we're very interested in, showed consistent expression across the lifetime. So potentially, as we think of therapies for postnatal life, this might be a target for postnatal therapies. And thanks to the BWF, I was actually really able to expand this study to include spatial transcriptomics data. So now we can start to approach not only, what are the targets in the brain, but then how does this really look for the whole organism? And because this fetus is so small, we can really profile the entire fetus at single cell spatial resolution. And so using this data, we're looking at our two targets, target X and target Y. And target X, we see that there is, less expression in the liver, which is good if we're trying to de-target the liver. But we do see some expression in the brain, so this is potentially one target that we could target. And then one target we see, specific expression in the CNS, which would potentially, again, allow us to target the cells, the brain specifically. And so what's even cooler about this data is we can actually look where Cree is or Cas9 or anything that we're really delivering because it's mRNA. We can pick this up in the fetus, and so what I'm showing you here is DAPI in blue, and yellow is the Cree signal. And again, the liver is obviously the most, where we see the most signal and very little signal in the brain. But even when we look in the brain, we can still see Cree signal, and then we can overlay our spatial transcriptomics data to see what are the specific cell types that are associated or correlated with Cree signal. And again, we see very similar things to what I show very similar to what I showed with flow cytometry data, but we do see some neuronal targeting, and what's really cool is now we can do this for the whole fetus and really start to begin to develop organ and cell type specific drug delivery. And so I then added a targeting moiety to my LNP formulation. And so I targeted receptor X with a specific peptide, and then I delivered this to the fetus, and then I looked to see how, whether it improved efficiency to the brain. And I was very excited to see that not only did it improve efficiency to endothelial cells, but it also increased efficiency to gene editing efficiencies to neurons as well as microglia, indicating that it actually improved transcytosis. So this might be an important receptor for not only transcytosis, but for LNP uptake. And with that, I would like to acknowledge, my lab, Bill especially, who's been a great help, my collaborators, and of course my funding.
Any question? I saw the hands over there. So.
Hi, how you doing? I really appreciate the talk. It was really amazing. I guess it's more of like a process, I guess, question, especially like working with like fetuses and stuff like that. Like, what is that like pipeline like working with like their like cells and stuff like that? How difficult is that? I'm sure it's very, very
very difficult. So I'm actually in the lab of a surgeon, so I got a lot of great training.
Yeah.
And so we do a laparotomy, and we're able to access the fetus, and we do direct fetal injections into the vitelline vein. I mean, it took me a year to be able to do those injections. It's very difficult, but there are benefits to the small size, and there are obviously downsides.
Oh yeah, sorry.
Really nice talk. For your SMA gene editing, did you look at the spinal cord at all to see like how many motor neurons are getting?
Yeah, so we did look at the spine, and so even though we saw like 3 to 4% of neurons in this, and it's even in the brain, it's even less in the spine. So that's why I kind of like the receptor Y, because it showed some targeting of the spine.
Thanks.
And they're actually not. Yeah, what we're seeing is they're not actually really that correlated, right? The CNS is not the brain is not the spine,
Right.
and which is pretty interesting.
One last question.
That was really awesome. My question is, it seems like you saw some type of disconnect between the survival studies and the protein expression, and maybe the transcription as well. Do you think that, like, what is the mechanism that you're seeing the benefit for survival for, and is it direct or related
Yeah,
to
so I
the
was
same
talking to
issue?
Dr. Merrill a little bit about this. There's potentially cross-correction, right? So if we correct something in the periphery, it might be trafficked from the periphery into the brain. I don't think that actually happens with the SMN protein because it's actually found in the nucleus, but potentially. But also, I think that there is a peripheral disease for these patients. I mean, they die from a respiratory disease, or they essentially stop breathing, and so we don't know really what they die so early that we don't really know what the other phenotypes are.
That's very cool. Thank you.